Welcome to Almost Here, Round the Corner Future Technology podcast with Richard Jacobs. Future technologies poised to transform our lives for better or worse are the focus of this podcast. Almost here means these technologies are now here and starting to be used, or just around the corner, from Bitcoin to artificial intelligence, 3D printing, blockchain, virtual reality, and more.
Hello, this is Richard Jacobs with Future Tech Podcast. Today my guest is Emily Leproust, CEO of TwistBioScience. Emily, how are you doing?
I'm doing great. Thank you for the invite.
Yeah, I'm glad you're here. First question is, can you tell listeners at TwistBioScience, what do you guys do? What's the company's mission and work?
Sure, yeah. So what we do at Twist is we make DNA from scratch. And to do that, we are using a silicon technology. And we are applying that technology to make DNA and advance the research of our customers in a few areas. One area is the production of chemicals by fermentation instead of using oil as a source of carbon. Another area is the modification of plants to get higher yield, better protection against extremes of disease, against extremes of weather, needing less fertilizer to grow food, so trying to increase food security for the planet. A third area is the development of therapeutics, vaccines, antibiotics to make the world more healthy. And then there are new applications of DNA, in particular around data storage. So the mission is to develop a DNA synthesis technology and apply it to some of the big challenges of humanity.
When you make DNA from scratch, are you attempting to make an entire gene from an animal, or how much DNA are you trying to synthesize?
Yeah, so in the grand scheme of things, those are relatively small fragments. So the genes that we make are lengths up to 3.2 kb, so 3,200 bases, so 3,200 letters. And if you look at the size of a human DNA, that is 3 billion letters. So we make about 1 millionth of a human genome at a time. So those are relatively small genes compared to the size of a genome. But that side of gene is what is being used right now in research to understand biology and to deploy applications that have a positive impact.
So you're making about 3,000 base pairs length, and is that a sweet spot in terms of different organisms, how they code for various proteins or other types of things? Have you seen why that length?
Yeah, so we started at 1.8 kb, so 1,800 base pairs, and now we expanded to 3,200 base pairs, and in the future we'll expand to 5,000 and 10,000. And so there's two reasons for that. One is a technology reason. The longer you go, the harder it gets to make DNA. The second reason is the genetic information is very relevant at that scale of a few thousand bases. On average, a gene is 1,500 bases, and so a typical gene is about that size. And if you add a promoter in front of the genes and maybe a terminator after the genes, that size goes up to about 3,000 base pairs. And then if you put two of those together to have a pathway or multiple genes, that's why you need 5 kb I don't know if you put 4 genes together, you may need to go to 8 or 10 kbs, and beyond that length, it becomes more difficult to model how those genes will work together. So it's a little bit less practical. So I'd say that a few thousand bases is where the demand is right now, because that's the natural unit of genetic information that our users work with.
Why is it harder to make longer sets of base pairs? What's difficult about it?
It's all hard. I don't want to say that making 1,000 base pairs is easy. I mean, making 1,000 is hard too. But if you use DNA less than 10,000 bases, you can use E. coli in the cloning process, and that is a very well understood, very well behaved organism in the most part. Once you want to go beyond 10 kb, you usually have to use yeast as an organism to help you manipulate that DNA. And then once you want to go beyond the 100 kb to the 200 kb and above, DNA becomes hard to move around. So even if you try to move DNA from one location to the next, if you use a pipette to do that, the DNA will shear. So moving DNA around becomes difficult as you go above a few hundred kbs. And then if you want to go very long, if you want to make a full genome, like millions of bases or even billions of bases, like in mammalian cells, the DNA is packaged around histones, which are balls of proteins, to help store the DNA, so that histone packaging is difficult to replicate. And then on top of that, DNA is also methylated in some regions to turn on and off some genes. And so doing methylation on large scale is also very, very complicated. So as you go from a simple genes to a pathway, to a full chromosome, to a full genome, and the level of technical complexity goes up and up and up and up. So as a result, there's a very large number of researchers that work on the gene level, and there's only a handful of researchers that are working on a very, very big system.
How do you feel about the human genome? That is 3.2 billion base pairs. Are you in awe of it now and the complexity, or you just said it's harder and harder to do longer and longer sequences?
Oh yeah, it's the biological systems are just incredible, and the pace of understanding and the pace of innovation is only accelerating. In the early 2000s, 15, 20 years ago, was the first time we sequenced one human. And it took several $1 billion and 20 years to sequence one human. And in the last 15 years, the technology around DNA reading, DNA sequencing, has changed so much that now you can sequence a human genome for $1,000. Soon it will be $100, and then it will be $10. So the ability to read DNA has changed dramatically, and as more and more reading has been done, the understanding of biology has changed considerably. But there is still a limitation around DNA writing, because once you understand, or you think you understand, you have to test your hypothesis, and you have to do experiments, and you have to write DNA to do that. And there has not been much changes over that same period around DNA writing. That's where Twist is coming in. Is we have a disruptive technology that is making DNA writing easier, faster, cheaper, and there's going to be a lot more understanding coming from it. So yeah, I am in awe of the human genome, in awe of its complexity, and even though we understand a lot, we still don't understand most of it, which is mind-boggling. But there's a lot of great biological discoveries to be made, and then use those discoveries to make the world a better place, make the world more sustainable, and make the world more healthy.
Right. So out of all your projects that you mentioned, which one is most exciting to you? And let's go into some more details of it.
I used to say that I love all my children, so they're all as exciting one or the other. So I'm happy to speak about all and any of them. One that lately has sparked the imagination of a lot of people is definitely around data storage. The idea that instead of using tape to store data for the long term, why don't we do the same as what nature has done? And nature is using DNA as the long-term media to store data. So in a way, nature has chosen DNA as its magnetic tape, and so there's definitely a lot of opportunity in that area to really change how data is stored in the long term.
Well, let's talk about the storage of data. I've recently did an interview on this. I don't know if the number is right, but it seems like DNA can store data 200,000 times as dense as any other current media we have right now.
I have not done that particular math, but yes, it's extremely dense. It may be even more than that. The latest number I have is that you could store 1 petabyte in a grain of salt. So you can go to the store and buy a terabyte hard drive, terabyte hardware. So 1,000 of those is a petabyte. And so with DNA, you could have a petabyte in a grain of salt. So that is extremely dense.
What allows it to be more dense than we currently can do? I mean, I thought we were running into fundamental limits of physics and running up against quantum effects and how we're storing data. How can DNA store it so much more densely?
Yeah, so I'm a chemist, so I am not a physicist, but in the semiconductor way of storing data, right now we are using scales of nanometers to make transistors and semiconductor chips, and that we're reaching some limits of how much smaller the traces can be. But DNA is on the scale of angstroms, so DNA is actually even smaller than the best that we can do on semiconductors by a few orders of magnitude. So it's just that DNA in itself is extremely small, and it's been evolved over billions of years. So there's been a lot of time to optimize and polish the ability that our cells have copy and make DNA. So I think intrinsically it's just smaller, and nature has a few billion years advantage on humans, and so that's why it's better. However, to be in full disclosure, it is not a fast storage. You need to be okay waiting a few hours to get your data back. So it won't replace a hard drive where you need the data in and out very quickly. But it will replace a catastrophic backup where you're okay waiting a few hours to get your database back, your customer list back, your information back.
Interesting. Okay, so it's right. So it takes a few hours, better for long-term storage. Do you believe that through engineering and silicon or other, however it's being done right now, that people will beat the storage capacity of DNA anytime soon and have it fast, or is it still a long way off?
You mean, do I believe that DNA would become a routine storage media in the short term? Is that the question?
Well, I guess you're saying it can't because it's very slow right now to read and write. Is there any way to speed it up so that it can be a lot faster?
Yes, there's ways to speed it up. Enzymes can process information very rapidly, and things can be done in parallel. However, even without the speed, even in any change in speed, it's still a viable media as a replacement to tape. Tape also takes hours to get the data back. And we've had a collaboration with Microsoft and University of Washington, and we showed that we could take data, encode it into DNA, make it as DNA, store it, and sequence it back and decode it, and you will get 100% back the bytes that you've encoded. So we know that as a media, it does work. And now the question is more a question of cost. Can DNA be cost competitive compared to magnetic tape? But from a time point of view, it is already comparable to the performance you'll get on tape.
Okay. Back to the crafting of DNA from scratch, as you put it. How do you do that? And is it a very difficult process to create it from scratch? And does that give you any insights into the origin of life itself?
So the chemistry of making DNA is well known. It was published in 1982 by Marv Caruthers out of University of Colorado. So how to stitch together A, C, G, and T, that chemistry is well understood, and is 35 years later, everybody is still using that fundamental chemistry. So it's really, really robust. The difficulty is in how do you deploy those chemicals on the machine so that you can not only make one piece of DNA, but make millions of different pieces of DNA in parallel. That is the engineering challenge. And yes, that is very hard. But if you apply the modern engineering method, and if you have co-founders like I have that are engineers and skilled in deploying those engineering principles, then you can build a machine that delivers the right reaction, the right reagent at the right location and do that at a very high rate. We do that hundreds of times per second, and build millions of different pieces of DNA in parallel.
Okay. Are you making RNA to read the DNA and zip it and unzip it, or what do you do with the DNA once it's made? How are you using it in various applications?
Yeah. So we are not application makers. What we do is we are DNA providers. So we enable people to develop applications. So what we do with the DNA is we ship it to a researcher, and then researchers use that DNA for their work around basic understanding of biology, work around storing data, work around the development of therapeutics, work around the development of bioproductions of chemicals or modifications of plants. So there's many different applications, but we don't do any of those ourselves. Instead, we do the hard work of making the DNA such that others don't have to do it themselves.
Okay, very good. Can we talk about one more application? Maybe you're again. I know you can't play favorites, but we talked a bit about storage.
Sure. Yeah.
What about curing disease? What are some applications or use cases for the DNA you're creating that are going to be used to help prevent or cure disease in people?
Absolutely happy to do that. And so maybe I can give a couple examples. The classic example is you have a disease and someone has done basic research to know that that disease is because a protein is not working well. So for instance, in the case of diabetes, you may have insulin that is not working well. So once the pharmaceutical company, once they know the protein that's the problem, then they look for a molecule that will bind that protein and restore its function. And restoring a function may mean increasing its activity, or may mean decreasing that activity. Both could be problematic. And so there are two kinds of molecules that are used to bind and have an impact on a protein. One are small molecules. Those are small organic molecules that can be derived from natural products. And in the past, all the therapeutics were small molecules. In the last decade, that has changed, and now most of drugs being developed are antibodies. And antibodies is what our own body makes when we get infected by a pathogen. And so drug companies, they try to mimic what the body does, and they test many antibodies to find which one binds the protein that they are trying to target. And so to do that, to make an antibody, you need DNA. And so they come to Twist, and they may get 100 different sequences, or 1,000 or 10,000 or hundreds of thousands of different antibody sequences. We ship it to them. They express those antibodies to make it, and then they have some binding assay to find out of all the antibodies they purchase which one binds better and which one either increases the activity of the protein or decreases the activity, depending what they want to do. So we are helping them do their drug discovery, find that antibody that will work well as a drug. And that is a very small part of the drug discovery process. Once you're done with the discovery, you have to develop the drug. You have to make kilograms of that antibody, and then you have to do multiple clinical trials, and then ultimately it becomes a drug. So some of the antibodies that we are making now that pharma companies are purchasing, in 10, 15, 20 years, those will become therapeutics that hopefully will save lives and increase quality of life. So that's one way that our DNA is used in the pharma. And then if you peek a little bit in the long term, our DNA will help really change how diseases are being addressed. In the past, it used to be blockbuster drugs. Everybody was getting the same drug. Now we are entered a time of precision medicine, and there's more advanced diagnosis to find which gene has a problem, and patients are grouped by gene diseases. And in the future, we'll have personalized medicine, where everybody will get their own personalized vaccines or antibody for their one mutation, and that DNA will come from us.
Okay, and then last question. What do you see as the future for Twist Bio in the next year or a couple of years? What do you think you guys are going to be coming out with?
Yeah, so I like to say that people usually overestimate what they can do in the short term, and they underestimate what they can do in the long term. I think for us, what we want is we want to be the catalyst and the accelerator of the research that people do to increase sustainability and increase health. And that means make DNA even faster, even cheaper, even longer than we can now, and make cloning obsolete in the sense of people will stop cloning themselves in their own lab because it will be more effective to get it from Twist. So that is the goal, is to really be an enabler and accelerate the pace at which our customers develop those life-saving drugs and better chemical productions for the environment.
Okay, very good. Now, what's the best way for interested parties to find out more about Twist and to start interacting with you guys?
Yeah, the best way is to go on our website, www.twistbioscience.com. Apologies for the shameless self-promotion, but it's the best way to learn about the products that we have if people are interested in being customers. We also have a blog section where we try to educate or provide information about how DNA is used and what are the positive impact coming from DNA. So we try to have a collection of information both for customers, investors, but also for the public to understand better all the good things that comes from DNA. Okay,
very good. Well, Emily, thank you so much for taking the time. I really appreciate it, and you guys are working on truly amazing stuff.
Thank you very much, and likewise, I really appreciate the opportunity to talk to you today.
You have been listening to Almost Here, Around the Corner Future Technology podcast with Richard Jacobs. Subscribe to this podcast, post a review to discover more future technologies that are poised to transform our lives for better or worse, such as Bitcoin, artificial intelligence, 3D printing, blockchain, virtual reality, and more.